Welcome to Analytically Delicious, Insights into Food Safety by LVA. Join us as we explore the latest developments, trends, and challenges in ensuring the safety of our food supply through analytical techniques, uncovering the science behind what makes our food safe and delicious.
Hello and welcome to our 5th episode of Analytically Delicious. I'm your host, David Steiner, and today we have the privilege of welcoming Dr. Elisabeth Varga, a distinguished scientist who uncovered the mysterious fish mortality event in Germany in 2022 through her groundbreaking research. As an assistant professor of analytical chemistry at the University of Veterinary Medicine, Vienna, Elisabeth brings over 15 years of experience in analytical chemistry with a particular focus on food analysis. Elisabeth's academic journey began with her doctoral studies at the University of Natural Resources and Life Sciences in Vienna, where she developed advanced LC-MS methods for detecting mycotoxins in food. She has held significant positions, including senior scientist and senior lecturer at the University of Vienna's Institute of Food Chemistry and Toxicology, where she made substantial contributions to advancing analytical methodologies and understanding toxin metabolisms in plants and animals. Her profound insights into identifying, characterizing, and quantifying mycotoxins and phycotoxins have earned her international recognition. Notably, her Erwin Schrödinger's postdoc fellowship at the Technical University of Denmark focused on fish toxic compounds produced by microalgae, highlighting critical connections between environmental toxins and the health of aquatic ecosystems. Today, we look forward to exploring Elisabeth's insights into her role in uncovering the fish mortality event in Germany and discussing the broader implications of her research for environmental and food safety. Elisabeth, it's great to have you here today. Welcome to our show.
My pleasure to be here today. Thanks for the invitation.
Elisabeth, doing research on aquatoxins in a non-maritime country like Austria sounds to me a little bit like investigating the social behavior of mountain goats in Denmark, right?
Yeah, that's a question I've been asked a lot, but Austrians love to eat fish and also marine fish, so it's important to have a valid supply chain. And the other thing is all the techniques I'm applying now for marine toxins, I can also apply for freshwater toxins. And in Austria, we have a lot of water, it's a water-rich country, and we might have some problems in the future, which I might be able to help solving.
All right, that's a very valid point, I understand. And what are aquatoxins actually? Why are they produced and what's their toxic mode of action?
So by definition, aquatic toxins are just toxins produced in the water or present in the water. Mm-hmm. And one specific group of aquatic toxins are the so-called phycotoxins. Mm-hmm. And these are toxins produced by microalgae. Mm-hmm. So very small microscopic organisms which are living in the water body. And the toxic mode of action is that they produce the toxins for their feeding. Mm-hmm. So they produce it to, for example, lyse other microalgae, also other small animals like crustaceans, and this is the primary mode of action. And that these toxins are also toxic for fish or humans is actually bad luck for us.
I understand. So how relevant are they in relation to food safety then?
So in relation to food safety, they are very relevant in case of mussels because they are filter feeders. And there it's mostly a problem if you go out and harvest the mussels yourself, because the ones which are available in the local stores or in supermarkets, there are monitoring programs in place to make sure that they are safe. And a lot of the aquatic toxins are actually not a threat to food safety, but more to food security, because we have limited resources available. So it's important that we use the resources we have. And if the fish are dying due to aquatic toxins, we cannot use them for food or feed purposes anymore. So it's, let's say, a waste of energy to produce this fish, which then cannot be used for food or feed.
Understand. So, Elisabeth, in your career, you have gained extensive experience in analytical chemistry, particularly focusing on mycotoxins and phycotoxins. So what motivated you to shift your focus to this area?
So my motivation was that I love mycotoxins, but I also especially love the technique I applied, so mass spectrometry. And since I wanted to stay at the university, I knew that I had to shift my focus. And I decided to do phycotoxins because there is a conference which is called Gordon Research Conference on mycotoxins and phycotoxins. And I knew when I go into the world of phycotoxins, I can still attend that conference and meet the old friends in the mycotoxin world. So that was one of the reasons why I decided to shift the focus of the toxin, but I stayed with the technique I love, so with mass spectrometry.
I understand. And what were some of the key challenges you faced in identifying and characterizing these different compound groups?
So the major problem in marine toxins is that we have a lack of knowledge. So very often we only know that the species is toxic, but we have no clue why it's toxic. So we have no clue about the toxin, why is it toxin, why is it produced, and so on. So we have a lack of standards. That's the first challenge, because as an analytical chemist, if you don't have a standard, you cannot quantify the toxin. The other problem is that the marine toxins usually occur in low concentrations. So they are really potent toxins, meaning that we have some challenges on the analytical side, because the bioassays are far more sensitive than the analytical methods we have. And one of the problems is the high diversity of the marine toxin, because it's not only one compound which is responsible, but in most cases we are speaking about compound groups, which makes it even more challenging as an analytical chemist, because we need standard for each analoga and each congruenter. Yes. And the other thing is, and I always find it a little bit funny, that we are talking about marine toxins or waterborne toxins, which are not stable in water. So they are produced in water, but they are not stable in water, which is actually a good thing, because I believe that if these would be really persistent toxins, we would not be here today, because everybody of us would have died.
Oh my God. So Elisabeth, in 2022, Poland and Germany experienced a significant fish die-off, resulting in a death of over 300 tons of fish. This incident was attributed to a combination of different factors, including unusual weather conditions, water pollution, and potentially toxic algal blooms. So given your experience in aquatic toxins, what role did you play in investigating and addressing this event?
Yeah, that was actually a really exciting time in my research career, because it was the middle of August in 2022, as you said, and really everybody was on holiday. And I planned to take 2 weeks focusing on writing a paper and being not disturbed by anybody else. But then it was the day after the holiday. On August 16th, I got a phone call asking me whether I know Prymnesium parvum. And actually, at first, I didn't understand the question, because the person who asked me pronounced it a little bit different. And so it took me a minute to realize, okay, he's talking about Prymnesium parvum. And then I said, yes, I'm working with this since several years, with this marine algae. And then he asked me, okay, whether I know the toxins produced by this marine algae and the prymnosins. And I said, yes, I'm analyzing them since several years. And then he said, okay, yeah, we have this incident in the order, but I've heard about it, and they now think that it might be Prymnesium parvum. And at that moment, I was a little bit skeptic, because Prymnesium parvum is a marine toxin. It's occurring in brackish water, so why should it be in the order, which is the freshwater water supply? And so I was skeptic. But I said, yes, they can send me samples and I can analyze them. And it was actually really, really fast, because on Wednesday afternoon, we had a phone call saying, okay, what we want to do. They send a courier overnight to Vienna, and at 8 a.m. I received the samples. I did the sample processing, and then at 6 p.m. in the evening, we knew that Prymnesiums are actually in those samples. So it took quite long to realize that it might be Prymnesium parvum, but since we already had quite a lot of knowledge about this microalgae and also the toxins produced by them, the analytical part was really fast. So it was 8 a.m. in the morning and 6 p.m. in the afternoon.
I understand. And what insights did you gain about its causes and impacts?
So I personally learned a lot about this incident because, first of all, sometimes it's important that you have to react fast and you should not listen to your lawyers because if you have to sign an MTA first before you accept samples, the other incident will be gone and you won't receive any samples. And the other thing is the scientific cooperation and especially the interdisciplinary cooperation was very important in this context and also the open-mindedness. And it was a little bit difficult in this case about the political situation which was involved in the incident because everybody wanted to blame somebody. And this was a little bit difficult also for me as a scientist because for me it's not important whom to blame. I just wanted to solve the problem and find solutions. And this was something I learned out of this. And the other thing is that, you have to be open-minded because sometimes you don't expect a compound in a certain matrix, but it might be there due to human interaction, due to climate change, and so on. So this is something which is really important in that context.
Yeah, very important also again to highlight how important scientific and interdisciplinary collaboration actually is. Elisabeth, beyond promethazines, what are some other environmental toxins that can impact aquatic ecosystems and how do they pose threats to the biodiversity?
So in the aquatic toxins, we distinguish between the mainly freshwater toxins and then the toxins which are more in the brackish water or marine water. And for the freshwater toxins, one of the most important classes are definitely the mycocystins, which are produced by cyanotoxins, which are blue-green algae. And they are also a problem in Austria, or will be a problem in Austria. So maybe you remember last year, in Ottenstein, it was closed and you should not go into the lake because of microalgae bloom. And the other thing are the marine toxins. There, for example, we are talking about red tides when the color becomes red due to the microalgae. And in this case, it's, for example, produced by Carenia brevis, which produces a toxin class of called brevetoxins. And these are highly potent neurotoxins. And one of the problems in this case is that they can accumulate in shellfish because mussels and shellfish in general, they are filter feeders, so they filter the water, but in this process they also filter out the algae and then accumulate the toxins, and that's why in the end it's then a problem for humans when we eat this shellfish because the toxins are concentrated.
Very interesting. What analytical methodologies did you actually utilize to detect and quantify these toxins in the environmental samples?
So in this case, we always use liquid chromatography coupled to mass spectrometry. Especially for the marine toxins, we go more into the direction of high-resolution mass spectrometry. And the simple reason for this is that we know the toxin classes, we know the approximate masses, but we also know that there exist a lot of different modifications and analoga. So we want to be a little bit open-minded, and the great potential of the high-resolution mass spectrometric methods is that you can look for known compounds, but in the same injection, you can also look for some unknown or some related compounds. And that's the big advantage compared to the triple quadrupole methods where you only see what you're looking for. And if you then expect or suspect that there might be some other toxins or modifications, you need a second injection for this. So that's the primary technique we use. Sometimes we do it in cooperation or in conjunction with diode array detectors, because some of the toxins also have conjugated double bonds, so you see them also in the DRD. which is then a second line of identification.
And what roles play bioassays in this relation?
Actually, bioassays still play a very important role. So, I mean, in former years, when we talked about bioassays in the microalgae world, we talked about mouse assays and taking really live mouse, inject the solutions, for example, from the shellfish, and then just see whether you see some toxic effects or that the mouse is dying or not. Now we will, of course, to animal welfare, we want to go away from the mouse assay to the in vitro assays, but it's still very important because the advantage of the in vitro assays is that we just look for toxicity. We don't look for the specific toxins, we just look, is it toxic, yes or no? And so we can also look for unknowns. And if we then, for example, have a sample which is identified as positive in a bioassay, we can apply all the analytical techniques for the suspected toxins. If we find them, great. If we don't find them, we can investigate further to find out which is actually the cause why we have cytotoxicity in this specific assay. So bioassays still play a really important role, but there is the shift from the mouse bioassay to the in vitro bioassay where we use cell cultures and not live animals.
Understand. And when we are looking forward, what are the key research questions or areas that need further exploration in understanding and mitigating the impacts of this specific environmental toxin group we were talking about?
So I think the most important thing is that it's a very interdisciplinary project, or there is the need of interdisciplinary projects, and also that we need more chemists, because really in the last years, the biologists were already working on that subject, and so we know these species are toxic, but we have no clue why they are toxic, whether it is just because of the high biomass, that they are using up all the oxygen so that the oxygen is not available for the fish or for the mussels, or whether they really produce a toxin which is then responsible for the effects we are seeing. So that's one of the things that we work interdisciplinary, combine our knowledge of these species is toxic, this isolate is not toxic, and then also for the chemists to identify, and then for the genetics or molecular biologists to try to identify the genes which are responsible for producing these toxins, so that we can, we know more and gain more knowledge about this. And then the last step, if we know, okay, what is the species responsible, we know how we can quantify it, and then we can go into mitigation and have some techniques or find some techniques which we can apply to either monitor the toxins so that we can have early warning systems, or ideally that we find techniques how we can prevent or either the algal plume or how we can stop the plume thereafter.
Elisabeth, beside your research activities, you are also working as an educator on the university. How do you engage and inspire the general public, especially children, to develop a greater awareness and interest in the field of analytical chemistry?
So that's a very important question, and it's a very important point for me, because I think as a university teacher, we are not only responsible for teaching the students we already have, but we also need to inspire children to pursue that path. And in this context, I think there are some great initiatives like the Long Night of Science or the Kinderuni, where you have the opportunity to show children what possibilities you have apart from the professions everybody knows of, like police, firemen, also lawyers. Every children knows what a veterinarian is, what a doctor is, but not a lot of children know what is an analytical chemist or what can you do in science in general. And it doesn't have to be chemistry, but it's also biology, microbiology, molecular biology, and that's something I think it's very important for the children to see what opportunities they had to have. And then the other thing is to really this enthusiasm for research, the curiosity, because children are really curious. Mm-hmm. And somehow during the school career, they lose this curiosity. And I think it's very important to keep this curiosity and also the persistence and the dedication to a specific subject. And so I think it's a really good initiative, this to like the Long Night of Science or also the Kinderuni.
Yeah, very important points, and I fully agree with that. Elisabeth, we are already coming to an end of this episode. So for the next generation scientists interested in chasing a career in analytical chemistry, what advice would you give them?
I think my biggest advice is to never give up because it might not always be a straight line, and there will always be some detours, and sometimes the process will be slow. But the most important thing is that, don't give up. And if you have a problem and you're stuck, try to break it down into smaller bits to solve these first, to then solve the big issue. Because also the Oder incident showed there are some small processes which are necessary to solve the big picture.
Understand and also agree on that. Elisabeth, thank you so much for joining us today and sharing your valuable insights. Your expertise and passion for analytical chemistry are truly inspiring.
Thanks.
If you found today's conversation as enlightening as we did and want to stay informed about future episodes, remember to subscribe to our podcast on Spotify or YouTube. By subscribing, you'll never miss a bite of our delicious insights into food safety and analysis. And for those of you with burning questions or lingering curiosities related to our today's episode or any other food safety topic, don't hesitate to contact service at lva.at. Your questions will be addressed either directly in our next episode or by our team of experts on our website, www.lva.at. Thank you for tuning in, and until next time, stay safe and stay informed.